Now, it's my pleasure to introduce today's speaker, Dr. Raja Mugasimangalam, CEO, Genotypic Technology. Dr. Raja is a veteran technocrat turned entrepreneur in the genomics sector since 1998. In addition to Genotypic Technology, India's first genomics company, he is also the founder of Qtilomics, India's first agri-genomics company. Applying genomics in real-life problems in research, product development, and testing are his expertise. He is member of Board of Studies in many universities for genomics, genome informatics, and bioinformatics, and of several professional organizations. He is also an active publisher and reviewer in peer-reviewed journals. He runs the world's largest genomics LinkedIn group for next generation sequencing and microarray with more than 1 lakh followers. Now, over to Dr. Raja for the webinar.
Thank you. Am I audible? I hope.
Yes, sir.
You are clearly audible. Thank you, Saroja. Thank you, everybody. This is, I don't know, the nth seminar in this series, I think 25th, which we started from COVID period. Firstly, thank you all of you. Totally 600 people registered for this webinar on epigenetics. Thank you all of you for also giving some information. We are having group heads, professors, PhDs, postdocs, graduate students, scientists, some clinicians, technicians, almost everybody. Most of you are from India. Some are from South America, APAC, some from North America too, Africa. Thank you from all over the world who has logged in for giving this information that vast majority of you are yet to start with epigenetics. I think now we are reaching almost 700 responses. This is 600 some few hours ago. So this is an important data to start with. Most of you are going to start. So it's nice and good, nice and good. It's good to start with the best thing than move. The best thing people are using the most widely used amongst you is a bisulfite and Sanger sequencing. That's you convert the DNA using bisulfite. I'll show you what happens when you put to DNA to bisulfite. The C becomes uracil, cytosine becomes uracil. And then when you do a PCR, it becomes T, thyroid. And then you sequence it, it becomes TA, CG becomes TA. Bisulfite and short reads here seems to be the ones which people are most using. Here you see nanopore. I'm glad 8% of you are using nanopore. MEDIP nobody is using. It's also another method. I think very, very little number of people. That's methyl immunoprecipitation. That's you only precipitate the methylated DNA and then you look at whether it got enriched or not. So this is the information which you gave. And here is about genotypic. We started in 1998. Sorry, there's a small bug there. And then those days it was microarrays, right? 1998 microarrays came in, printed microarrays, then came the NGS here. So this here, first iron torrent, and we certified for iron torrent, Illumina CSP, everywhere we joined. Then came Nanopore. The Nanopore CEO was in India, and we got really impressed And 2015-16, we started using nanopore when it was in early times. We became nanopore certified service provider, and also now a channel partner. We do a lot of R&D with on nanopore, with nanopore. And now we distribute nanopore and many other products along with it. We also have a big lab with nanopore sequencer. Small ones, big ones, everything is there for you to use it. And then we have now this 2023 or 2024 here, PacBio Onso sequencer, short read at Q40, Q50. So why short reads? Well, the advantage is you can generate a lot of data. Q scores, I think still short reads are useful while everything is moving to long reads. And as you know, nanopore you can do high accuracy base calling, ultra high accuracy or super high accuracy base calling. Everything is possible now. Our expertise is in genomics, not just in genomics, how to plan the experiment, conduct the experiment, how to analyze it, bioinformatics. We also have what we call as bridging the gap. New technologies come, biologist problems still remain. Can we answer new questions? Rather, can we ask new questions? So today's talk is going to be on, can you ask new questions on epigenetics? Because genetics, a lot of things have been already done, right? So we are a team of around 75 people, around 12 masters, 30 masters, PhDs. We do a lot of R&D, product development and new technology development. More than 650 publications, a lot more coming, 650, what we know. And then we have two sister companies, Dithiomics, which works on diagnostics. These are all cancer panels, genetic disorders, or anything you say, applying genomics or NGS in diagnostics. Dithiomics work with clinical samples. And then Qtelomics works on solely on agriculture related projects. So we do lots. Very briefly, this Q40 plus Onso is something which we have started now, but basically lots and lots of nanopore sequencing. Many of them are applications or questions which never had, like you can sequence RNA itself. You can sequence DNA, native DNA without PCR or any change, right? No chemistry, it's only physics. And last but not least is this protein assay, SomaLogic. You can look at protein expression, almost as good as RNA expression. So let's come back to epigenetics. What is epigenetics? We all know there is 4 bases. The cytosine gets methylated. 5-methyl cytosine, right? So this 5 is role, lots of role in that. Not just 5-methyl cytosine, there are different cytosines. So one, the importance of methylation is the phase escape restriction or restriction nucleases attack the methylase signature. That is, if you are not methylated on site, the equivalent restriction enzyme will go and hit that GAA-TTC. The very first restriction enzyme known is on the ulta, on the reverse. That is, a phage DNA is methylated, which actually helps it escape from all these T-even phages, T2, T4, T6. The phage DNA is methylated to escape restriction nucleases. But there is another restriction nuclease which actually hits the modified DNA, methyl cytosine DNA. That is the RGLA, RGLB, or now called as MCRBC, MCRA. I did my PhD in these systems. This is what was prohibiting a lot of plant DNA and human DNA to be cloned. So all the E. coli, the strains which are lab strains, or RGLA, RGLB minus, or MCRA, MCRBC minus strains, so that it won't attack methylated DNA. So you can clone DNA from plants and humans. And then there is DAM methylation, DNA adenosine methylation. See, this DAM methylation is used by bacteria for DNA repair or error correction. For example, a DNA is being replicated. So the newly replicated strand or the synthesized polymer by polymerase might have a mutation. But how do, and then the two strands are made with one being mutated, one not mutated. So how do you know which one to, what to correct against what? Right? How do you correct? So this adenosine methylation happens after a certain period into the second strand. So when the second strand also gets methylated, it don't know. But until then, it knows which is the first strand and which is the second strand. Got it, no? So this DAM methylase is used. So methylation has lots of applications apart from these two, but this is what we know. So how methylation looks, let me wear this little cap. It's like this cap. You wear this cap. I'm the same guy, but I'm wearing this cap. But unlike this cap, once a DNA gets methylated, it remains methylated. It's not reversible that easily. It sticks like Fevicol. You put it and it's stuck to you. Even when the next strand is synthesized, the DNA methylases have specificity towards hemimethylated DNA. It will run and methylate the next strand also. So once methylated, so what you see here, it's methylated for good. So this is how when people age, DNA gets methylated and you never lose that methylation. Some regions, methylation is lost and you never get it back in higher eukaryotes. So why do we have to investigate methylation? First and foremost reason is methylation is important for gene expression. Very plain and simple. Whenever there is a methylase, the polymerase, it works like a speed breaker. Right? If lots of places it's methylated, that's what we call CPG island, then the polymerase even falls down. There will be zero transcription. If a little bit of methylation is there, transcription slows down, or the number of transcripts made gets reduced. So almost all the eukaryotic and many prokaryotic are regulated by methylation. And this methyl C plays a role on it. And also this also is used by histone modification and others to pack it. So if the DNA is not at all methylated, if it's completely naked, it gets completely into a euchromatin kind of a region. So methylation is indeed important. So here is what I said, aging. Globally, certain regions get hypomethylation and hypermethylation occurs selective regions. So over age, certain genes never gets expressed or it gets keep on reduced. And does epigenetic play a role in plants? Of course, it plays a role because plant DNA are heavily methylated. Also, there is a term called epimutation. Epimutation means an abnormal methylation. So lots of systems are there to study it. In Arabidopsis, here is a paper where once this methylation happens, that epimutation happens, a change in methylation pattern, it's retained. So what is interesting is people thought epigenetics is not inherited. But now we know epigenetics also gets inherited. Sometimes you get a chromosome from father and from the mother, a methylated from a X chromosome continue to get being methylated. That's also a way of silencing, right? One of the X chromosomes. X chromosome, only one X chromosome gets expressed, not the other one. So you can see that epigenetics is really important. It's not, we could not ask many questions because there was not good way to study. So how was methylation, DNA methylation studied? Earlier, it was done using liquid chromatography, electrophoresis, microarrays were used. And of course, here you see the first generation sequencing or the Sanger sequencing. You take a DNA, you treat it with bisulfite, and you have another one without bisulfite treatment, and then you do PCR amplification. I'll show in the next slide what happens during that time. It's called bisulfite conversion. You convert all C into U, and the U becomes T when you do the PCR. And when the second generation sequencing came, this Illumina short reads, Thermo Fisher, and all of them, now MGI is also there. All of them used the same principle, convert by bisulfite and then sequence. There are lots of problems in doing this. First is you can only identify methyl cytosine. Second, because the DNA complexity gets reduced, because all or wherever it's methylated, C is becoming T, alignment, all those issues coming wherever it's methylated. So that is an inherent problem for it. Now, the new gold standard, nanopore, has come, wherein there's no chemistry, it's only physics or electricity. The single-strand DNA goes through the pore, electric signals are shortly. So this is becoming a new. So this is the bisulfite conversion method. Once you treat with bisulfite, all the methyl cytosines, I mean the unmethylated cytosines, become uracil and it becomes T, whereas the methylated cytosines remains as a C. So how does nanopore? Very briefly, in nanopore, the DNA, RNA is ligated. It takes only 5 minutes to do that to a motor protein. It's nothing but a helicase. And the helicase unwinds and pushes it through the pore. And between the pore, or the two ends of the pore, there are electrodes which can make the electricity. So when then there is no DNA, you have good voltage going. When the voltage goes down and then you see the wave is the shadow of the DNA in the electric field. That's the file you get. So electric signal becomes the wave, and that wave gets converted to ATGC. Since it's a shadow, if there is a DNA, if the cytosine or adenosine is methylated, that can also be captured. So this modification can also be identified. So now it becomes very, very simple, right? There is no bisulfite, there is no this, there is no bioinformatics, this, that, and all. Now you can plain straightforward read ATGC, and then is the C methylated or not. Or long read, I can actually look at some genes in one chromosome, it's not methylated. In the other pair, it's methylated. So we can say, hey, these regions are shut down in one chromosome, these regions are open in another chromosome. That much we can say nicely. So why should we use nanopore for methylation? First is, when you have short read sequencing, you need longer X coverage to get the data because lots of things are lost also. And then the number of CPGs called is phenomenally high with long reads than with the short reads. And more importantly, we can look at 6-methyl adenosine, 5-methyl C, which is the one which is identified by the bisulfite conversion, 4-methyl C, or anything, even any other methylation or modification of the DNA, one can see it. So this is much more easier, and then it gives a lot more detection of lot more changes than methyl cytosine. So there is no PCR bias, there is no GC bias when you do nanopore for methylation. And the long reads also increase the mapping on repetitive regions and CG-rich regions, GC-rich regions, which short reads struggle to do. And that's where actually you have the gene regulations happening, right? And then long-range epigenetic patterns can also be seen. This whole chromosome, this copy of the chromosome is methylated, that copy is not. This is called phased reads, which is not possible with short reads. So you can see which regions where those can be seen, and you can see the and then there is also people before who are doing whole genome and also targeted methylation.
Class, beta class
Whole genome by reduce representation by sulfite sequencing. That is by using only a small portion of the genome by using enzyme and then do bisulfite and only sequence a part of it. So now targeted methylation and direct methylation with ONT we can do, which is more called as a reduced representation, bisulfite, or what we call as adaptive sequencing, where you sequence more of regions of interest and less of regions of non-interest. So this chart clearly explains how you can look at more different types of methylations. So this is called adaptive sampling, in which when the DNA is being sequenced, in one, as I told it, in 1 second it reads 400 bases. So in 1 second itself, it can say whether it is a region of interest or of not interest. So it can stop sequencing and sequence only regions of interest in the other poles. So this helps actually not to meddle with selecting different fragments by restriction enzymes or by other methods like sure select or other methods where by binding you can pull out or capture regions of interest. So this makes life easier. It's all only happening in the software or in the sequencer. So here is where it is. If regions of not interest, the strand is rejected. If the region is of interest, it continues to sequence. So this is called adaptive sequencing. So it easily gives 5 to 10-fold enrichment, which means for regions of interest, you can do lesser amount of sequencing itself, like in one flow cell. If you're sequencing 2 genomic DNAs, you can now do maybe 10 genomic genomes for methylation. This is called PCR free targeted sequencing. And there is Sure Select. This is done by Agilent, Twist. Many other companies are making this. So here you pull down the regions or use bisulfite convert, pull down the regions, and then you sequence it. One of the important things here is what you will miss is only regions of interest you are sequencing, not the rest. But even within that, you are not getting everything. But since this was the gold standard, it was compared with the nanopore whole genome methylation or a targeted methylome. So we could see that it's all 99.9% overlapping data between the two and in the common regions, but there is a lot more on nanopore. That's very important. Nanopore is finding the same methylations which bisulfite conversions find, but there is a lot more it can find. So that's something surprising. So we ask the question here, who is the gold standard now? There's many other applications. The cell has lots of promises, right? Cell-free DNA is kind of non-invasive. You can take your blood, look at DNA in the serum or in the plasma, right? So here we can look at biomarkers for a cancer screening tool, or you want to prognose something, or which will be the therapy, right, to do it. You need not go and surgically remove some parts. And this can be used for looking at the fetal DNA in the mother. So here the whole process, sample preparation, library, sequencing, all can be done very quickly. And the library preparation is easy. There is no PCR. It is just adapter ligation or use a transposon and make the library. And you can barcode up until 96 samples directly. And then you can start sequencing. And then you can look at how much input DNA and how much is getting aligned. And in these cases, if the fragments are small, you don't use a rapid transposon-based library, but use ligation-based. DNA methylation is because the adenosine methylation and all of them, people only use the restriction nuclease or modification sites. That was the original idea. Then methyl cytosine, of course, 5-methyl cytosine, you can use bisulfite, but there is so many other methylations, changes happening on the DNA. All of them can now be sequenced using and analyzed. While you sequence the genomic DNA, you can look at the methylation also, technically free of cost. Just sequence the bug, but now you look at the methylation. So these methylation signatures have lots of applications, whether because, as you know, the methylation signature is also inherited for a certain number of generations, right? So we can say, hey, this bug, though it's almost identical to the other, this is coming from this visitor who came from abroad. This bug is the progeny of that, whereas that is the progeny, it's a local bug. There's lots of applications of bacterial DNA methylation. So here also the step is you align, get structural variants, SNPs, that is one side, and then different methylation events can be detected. So now coming to methylation analysis in the plants. As you know, plant DNAs are the most methylated DNA, cytosine methylation, to the level that it's almost hard to get many regions of the plant cloned until this MCRBC, all of these things were identified. So these are experiments, actual experiments done at Genotypic. We are doing some projects at Qtelomics also. So here, after nanopore sequencing, see here there are many different base callings which we can do. And then we use, this is the bioinformatic pipeline. It's not as easy and straightforward like you have to run through different programs to look at different methylations because these are all models. How the base calling, converting the wave back to the DNA sequence, we have to use different tools. So that itself takes quite a lot of time. So once you have the differential methylation, then you annotate for CPGs or promoter regions and say, probably these genes are upregulated, these genes are downregulated. But for that, you have to do gene expression to double validate whatever is the finding here, or some of them. Because as I said, no, methylation leads to reduction in transcription, right? Or complete shutdown of transcription if it's heavily methylated. So this is how the methylation data is represented chromosome-wise, which regions are hypermethylation, hypomethylation, whether both strands are methylated, single strands are methylated. So all of these are now gaining significance. There are companies which are only working on epigenetics. They are no more looking at genes or SNPs. They are looking at, can I take these seeds, treat it, maybe I can make them drought tolerant by methylation. So there's lots of publications. As I told, no, epi mutation is one thing. That is the aberrant methylation patterns is happening. There are quite a few human diseases which are connected with aberrant methylation pattern. But here are the examples. This is epilepsies. This is cancer, blastoma, Helper cells. So here is kind of history when it started with methylation sensitive restriction enzymes. One Dr. Michael McClelland, with whom I did my postdoc in San Diego, he was publishing a database of methylation and sensitivity of restriction enzymes. Sensitivity and, of course, resistance to different restriction enzymes. That table was used, New England Biolabs used to use that table and sell enzymes. So this methylation pattern, please use this restriction enzyme. And then they were selling also the methylases. Otherwise, methylases have no use so that they can make control. And then obviously HPLC, you can digest the DNA completely into single nucleotides, run it, and then you see. And then came here this 5-methyl cytosine. This is anti-5-methyl, means it's an antibody. So methyl cytosine antibody, or then what is called as MEDIP, was used. PCR, and you can see this Maxim Gilbert sequencing was actually ideal for DNA methyl sequencing. We are all use Sanger. Both of them, as you know, got the Nobel Prize for sequencing. Sanger was used. And then this HEPA2 PCR assay, because HEPA2 is a methyl specific restriction enzyme. So you digest with HEPA2. There is another pair of enzyme which does not digest, which digests both of them, combine and do that. And then comes the bisulfite changes here. And then you can slowly see here, then the microarrays came where what we call as MEDIP was used. This is simple way. You chop the DNA, genomic DNA into small pieces, and then take one fraction, which is called input DNA, which is as it is DNA. And the other fraction is you immunoprecipitate with the anti-5-methyl cytosine antibody. So you're enriching wherever it is methylated. So you will only know what extent it got methylated, those regions, not specifically how many of them. Then came here, when NGS started coming up, then the bisulfite conversion, library preparation, sequencing, that became the gold standard because it was very easy to do. Even now, if anyone asks gold standard, because after PCR, the C becoming T, right? So that's what is called as the gold standard. Then the short reads took over. Now the long read nanopore sequencing is becoming the new gold standard. I would rather say has become the new gold standard. People are only now trying both of them just to say, hey, I'm getting the same thing. I'm getting more, but now I'm believing that more also. So there is a shift in this new gold standard now currently happening. nanopore users, publications, how they benchmark the nanopore methylation from the publicly available bisulfite data sets. So here, this involves hundreds and thousands of samples, because for nanopore, you're not going to do anything special. After sequence, whenever you have the sequence data, you can look at how many are methylated, which regions are methylated, because you have to only base call in a different way for the methylation. So what it reveals is there is lower bias in nanopore sequencing, and that higher mapping rates, because anyway the reads are mapping, because there is no modification, because we want, unlike C changing to T, right? And then greater reproducibility of the same sample. Bisulfite conversion sometimes happens full, sometimes it doesn't happen. And then it's fast analysis. First round itself, you get the answer. And I'm just repeating that slide for you to know slightly more how this works. The DNA, RNA are ligated to the motor, which is the helicase, which pushes the DNA through the pore. It runs at around 400 bases per second. And the electrode kept at the two ends of the pore now measures the electric. So as I told, it's like this. When there is no DNA and the DNA enters, you generate the wave, which is the number in the electric field. So wherever it is methylated, you will see that also. So from electric signal, you get ATCG, and base modification can also be identified. Basically, any base modification. This is very, very important. Any base modification, all of them, you need some controlled DNA, either chemically synthesized, which they have already used, or naturally available, to know which methylation is there. And now you can go back and find out what is methylated, or the whole methylation pattern can be deduced. So to summarize, epigenetics is simplified. I would say not just simplified, is much, much more simplified. Actually, it's not even a different reaction. You're doing sequencing, boss, that's enough. Now you can look at methylation also. It's not another reaction where you have to go and do bisulfite convert sequence and look at it. Same sequencing reaction. So that's something, a big blessing. And this is, I would say, the new gold standard. So how we can help you? You can send the samples to Genotypic, hundreds of them, few of them. You have to only have the right biological question, right? Something which is not describable by genetics can be described by epigenetics. You can send the samples to Genotypic, and within a few weeks, we can return back data chromosome-wise, which are the regions which are methylated, hemi-methylated, and what is the difference between the two samples. Sometimes it could be age. We have already done that over age, which regions gets methylated, non-methylated. Same person at two different time points, like 5 years ago or now, or cell line, or between people, between siblings. And methylation can also be used when you take twins, identical twins, and look at the differences in epigenetics. Now you can say what are the genes which are controlled by epigenetics, right? That's a twin studies ideal to use here. Or if you want, you can acquire the nanopore sequencer from us. Within a month's time, we can install the sequencer and show you how to use. So thank you everyone for listening. Thank you, Sudha, Saroja, Meenal, and Kirti, and Abhinaya, and all for inputs and help to make this presentation. You can scan this barcode, you will go to our site, or you can email to our folks. If you're outside India, global, you can write to Kirti. If you're from corporate, Bharat, academics, Swapnil will be happy to talk to you. So thank you, everyone.